Hi everybody, this is Dr. Nadia Fouladian. We're going to start chapter 27 and we start with the male reproductive system for this video. So for the reproductive system in general is designed to have a specific function, right? The main purpose of having a reproductive system is what? To reproduce another human being, perpetuate the species, right? The reproductive system produces, stores, nourishes, and transports the gametes. Gametes are the sex cells. That is as we have it here. The male produces the gametes, we call them sperm, and in the female, we call them oocyte, right? So these are the sex cells. What is different between these cells and all the other cells of the body? All the other cells that we call them somatic cells, they have 46 chromosomes. These sex cells, each one have 23 chromosomes. And when they combine together, make a cell that has a 46 chromosome as all the other cells of the body. And that is the one that we call it a zygote, as you see that here. And that's due to a process that we call it fertilization. Joining of a sperm cell and an oocyte is called fertilization. And fertilization results in the formation of a zygote. So then after the zygote, that is a cell, one single cell with 46 chromosomes, start dividing. So it goes under mitosis, right? Then becomes double. Those each one get double until it just starting developing and differentiating the different layers and all those things to make the fetus, right? So that's a discussion of the embryology. Here we're going to talk about the reproductive system, how they are producing these gametes, which organs are taking part in the formation of the sperm in the male and oocyte in the female. We'll go over all details about these ones and all the organs that we have that we need them all to have not only producing the gametes but also have all other sex activity and characteristics to make the whole things happen, and these sperm and oocyte combine together to make the zygote. Now, for the reproductive system in general, that applies to both sex, but then later we're going to talk about each one in details, right? We have gonads and we have some external genitalia. Here we're talking about so gonads are the organs that are making the sperm and oocytes. In the male, gonads are testes, in the female they are ovaries, that they are producing gametes and also they are producing the sex hormones. Right? For besides the gonads, we have the reproductive tract, some tubes, tract, passageway that take these sperms and oocytes and get them to the proper place that they need to be, right? Ducts that receive, stores, and transport the gametes. Then we have some accessory glands that they have some secretions that we need them. They secrete some fluids, and inside of those fluids we have some material that are necessary for those gametes, sperm and oocytes, to remain alive and to remain functional, right? Then we have the external genitalia, the external part of the reproductive system, that we have them in the perineal area. Those are perineal structure. You remember the perineal area is the area between the urethra and the anus, right, on the bottom. Right. Now, let's start with the male reproductive system, and after this two videos that we're going to have for the male reproductive system, we continue as the female reproductive system. So for the male reproductive, the parts that we have, They include scrotum. That's a pouch of the skin that we have around the testes. Testes, as we discussed, this is a gonad that is making the sperm, and also they're making some sex hormones for the male. Inside of the scrotum, we have another organs of the reproductive system that we call it epididymis. Epididymis and testes both are located inside of the scrotum. We go all over all the details about all these organs. Then we have a duct that after epididymis, we have a duct that continues as the ductus deferens. Sometimes we call it vas deferens, VAS. Then urethra in the male also, besides being part of the urinary system that brings the urine from the urinary bladder towards out of the body, it also is a common passageway for the sperm too. Sperm that is formed inside of the testes, then it goes into the epididymis, then it goes to the ductus deferens, and then from that joins the urethra, and from urethra goes toward out of the body. So as you see, the urethra in the male is part of the two system, urinary system and reproductive system. And again, as we discussed, we have some glands that they have some secretions. The ones that we have in the male, we have one seminal gland, prostate gland, and the other one we call it bulbourethral gland, right? We're going to talk about all these ones, which secretions they have and what are their functions. And finally, we have as the external genitalia, another part besides the scrotum that is outside. Here we have the penis. Now let's go and review all those ones one by one. So here you're looking at the sagittal, mid-sagittal sections of the, almost mid-sagittal section of the male human, and you see that all these ones have been cut in mid-sagittal, but you see these tube have been shown parasagittal, and also these parts of the scrotum, that this is this pouch of the skin that we have outside of the body, and also this part that is testis, that's epididymis on the top of that then continues as the ductus deferens or vas deferens that you see that goes all the way here. These are located in parasagittal to the side, but they have been drawn here in this way so that you can see the relation of their organs together. So again, reviewing all the parts that we have the external genitalia of the penis, we have this prostate gland that you have it here, and you have the urethra that, as you remember, the first part of that passed through the prostate gland, prostatic urethra, then it has membranous urethra, then we have a spongy urethra. We're going to review those ones again. You have another gland here, bulbourethral gland, that we're going to talk about that. Another gland here is seminal vesicle or seminal gland. We're going to talk about these more. And also, as we discussed, you have the vas deferens or ductus deferens, you have the epididymis, and you have the testes, that these parts of them are located inside of the scrotum, right? Now let's go talk about each one by one, starting from the scrotum. So we have two testes that hang inside a pouch of the skin that we call it scrotum. So pay attention. Testis, T-E-S, T-I-S is singular. Testis is plural, right? But the pronunciation is very close to each other. So the scrotum is outside of the body. Testis or testicle size is about 5 cm long, 3 cm wide, and about 1 inch, 2.5 cm thick. And it weighs about 10 to 15 grams each, right? And as we said, it's located inside of the scrotum, and scrotum and testes are located in an adult person, are located outside of the body, as we see that here, the location of the testes is in the scrotum and outside of the body. Now we have a process for the testes that we call it descent of the testes. When the testis is formed during development, it forms inside of the abdominal cavity and the location is near the kidney, right? Then, as the embryo develops more, these testes going down and finally gets out of the abdominal cavity. Now let's go review that one, how this process is happening. We have a kind of ligament-like structure that we call it gubernaculum testis. That one holds the testes attached to the wall, right? In the place that it is. As body growth proceeds, gubernaculum testis doesn't elongate. So I will show you the pictures and you will realize this more. That is the key in the process of the descent of the testis. Gubernaculum testis pulls testis inferiorly. So you will see that it is not that this one, gubernaculum testis, is not pulling the testis down. It is that it keeps the testis in the original position that it was. And as all the other part of the body are growing, then it remains in the place that it was, and therefore it seems that it's going down. Testes descend toward the scrotum by passing through the inguinal canal. I will show the route to you. And the ductus deferens, blood vessels, lymphatics, and the nerves, all the things that are related to the testes, remain bonded to the testes in the cord that we call it a spermatic cord. So spermatic cord is all these structures that are related to the testes, and they are all coming down from the original position that is near the kidney, and then go all down, get to the outside of the body, inside of the scrotum. So pay attention right here that, as we mentioned in the urinary system too, that the blood vessels of the testes also formed near the kidney. And when they go down, or the testis goes down, the blood vessels, the origin remains over there, that you will see that they, like one of them at least, like one gonadal vein comes from the renal vein or joins the renal vein, and then continues all the way down toward the scrotum in the neck. So that is the reason that these gonadal vessels, they go all the way up near to the location of the kidney. Now, regarding this descent of the testes, why do we need the testes or testicle to be out of the body, right? There is a reason for that. The testicle development requires a temperature of approximately 98.C degree Fahrenheit, right? It's almost 37 degrees centigrade, right? And we need that temperature at the time of the development of the testes. Therefore, the development of the testes is happening inside of the abdominal cavity, that we have the core temperature of the body, the usual internal temperature, that is 98.C degree Fahrenheit or 37 degree centigrade, right? But after the testes develop, then when we want to start development of the sperm, it requires a temperature of approximately 96.6 degree Fahrenheit. So it's about 2 degree less, 2 Fahrenheit degree less than the core temperature. Therefore, it has to grow outside of the body. The development of the sperm cells happens outside of the abdominal cavity. What does it mean? It means that if the testis in the original time, at the beginning, is outside of the body, the structure of the testis cannot be formed completely. And if it doesn't come out and remains inside after it develops, it cannot produce proper sperm, right? So that's the reason that we need to have this descending of the testes to be formed inside of the abdomen and then to get out of that at the proper time. Here you see the picture that shows that the embryo that you have in the 2-month development. You see this part of the body from the location of almost near the kidney all the way down is about 5 millimeter, half a centimeter. You see how short it is. And if you look at here, you see that you have a testis here, one single, and you have the gubernaculum testis that is attaching to the testis to one end and attaching to this part of the body on the other end, right? Now, it becomes 3 months, 1 month later, the body of the embryo and the fetus is developing, and you see that the scale of the 5 millimeter now is not covering up to here, is about less than half, right? But still the length of the gubernaculum that we had from the beginning is still 5 millimeter, right? Now it seems that the testis is going down. That is actually this gubernaculum keep the testis in the position that it was 5 millimeter further from this part of the body. And now it continues, the growth continue, and at the time of the birth, that 5 millimeter gubernaculum that is still we have it the same size, now it is outside of the body. So this is the way that we see that the testis from inside of the abdomen is actually goes all the way down toward the sperm. Now in this another picture, you see the structure that are developing the reproductive system besides the one that they are developing the urinary system. You see the location of the kidneys, the developing kidneys at the 2 months of the embryonic period, and then you see the structures that are going to make the reproductive system, right? The kidneys remain in their place and the fetus and the embryo and fetus are growing, right? So these are getting bigger and developing more and all those things, and at the same time these other structures that are related to the reproductive system are developing more and growing, but as we said, gubernaculum keeps them close to this part of the body. And at the 4 months, they are in this level. When it becomes the 7 months, they are passing through the inguinal canal. The inguinal canal is a canal that we have it here. In the inside, you have an opening toward the abdominal cavity. In the outside, you have an opening toward the outside here. Then, by keeping this distance of the gubernaculum testis as a stable size here, this pulls all these ones through the way that it was here. And finally, at the time of the birth, the testis has come out to these locations. Pay attention that sometimes at the time of the birth it may not be completely out, but this is in the process to getting out. They may give it a little bit of the time to get out, but if it doesn't happen, they have to do the surgery and bring it out to descend out of the testis. Now, having all these ones in mind, just showing you these all the structures that coming besides the testes out, they are located in a sheet of a connective tissue, and all together they called a spermatic cord. Now let's see what do we have inside of the spermatic cord. Inside of a spermatic cord we have the ductus deferens, that is a tube that takes the sperm after the epididymis, take it toward the outside that I have shown. Then we have blood vessels We have several blood vessels here. We have differential artery, that is, we call it differential because this is going to supply this difference. Because of this difference, we call it differential artery. We have testicular artery, that those are the gonadal artery. In the male, we call it testicular artery. And we have a pampiniform plexus of the testicular veins. Testicular veins makes a plexus, a network of the vein, that they are helping in the control of the temperature around the testes. As we discussed, having a stable and lower temperature compared to the core temperature inside of the body is essential for the sperm production inside of the testes and its functions. Therefore, these pampiniform plexus are helping in the cooling down of the temperature or all those things, right? Then we have some nerves, that the two nerves that we have here, the ilioinguinal nerve and genitofemoral nerve. These are two nerves that we have in the spermatic cord, and you need. And that spermatic cord passes through the narrow inguinal canal that connects the scrotal chambers to peritoneal cavity. Peritoneal cavity is the cavity that we have inside of the abdomen, as you already know, and scrotal chamber is the space that we have inside of the scrotum, right, that we're going to talk about. Here you see in this picture, you see the spermatic cord. You see that this is the testicular artery and vein that have come from near the kidney all the way down. Now they have passed through this inguinal canal. This is the internal opening here, and you have the external opening in this side, right? Have been cut so that you see all the length of all the structures that you have inside of the inguinal canal. Then you see that inside you have the testes. You have epididymis, then it continues with the vas deferens, and when it gets passed through the inguinal canal here, then it looks back toward the urinary bladder, goes behind the urinary bladder. And here you see the pampiniform plexus here, and you see the arteries and nerves. Right, these yellows, right, and you see these arteries. So this was regarding the spermatic cord, that you have all the structures. Now let's talk about all the layers that we have around the spermatic cord, that they are covering all these ones until they get to the skin, that is the scrotum. You have several layers here in the wall of the scrotum, and they are covering, and you'll see what is their function. So for the scrotum and the positions of the testes, scrotum divides into two chambers. Each one is called scrotal cavities. So there is a septum between these two chambers. The chambers are separated by the raphe of the scrotum that can be seen as thickened tissue that extends from the anus across the scrotum and along the anterior surface of the penis, that I will show you. This is outside of the body, right? But inside, we call it a scrotal septum, the one that divides the two chambers. Inside, we call it a scrotal septum, that makes the two scrotal cavities inside, right? But from the outside, we have raphe of the scrotum. Then we have a layer, the first layer that we call it, the first layer around the testes, that we call it tunica vaginalis, that lines each insides of each cavity. And this tunica vaginalis is a serous membrane that lines the cavity and reduces friction between the opposing outer and inner serous layer. Let's go and see the positions of them. As you know, insides of the abdomen have been lined with the peritoneum, parietal peritoneum, right? Now, as the testis is descending and is going to come from the abdomen, goes into the inguinal canal and goes outside, it takes one layer of the peritoneum with itself, right? And is as it's going out, that and it makes a double layer here. And when it goes down here, that double layer around the testis, one is covering around the testis itself, that we call it visceral. That layer of the peritoneum is the one that is making the tunica vaginalis, right? Tunica vaginalis makes one layer around the testes and epididymis, that's the visceral part of that, and makes another layer that goes and covers inside of the lining insides of the scrotal cavity here, and that's the parietal layer of the tunica vaginalis. So we have always one layer of the tunica vaginalis around the testis. That is like a peritoneum anywhere else that you have seen. You can cut that and you can separate it from the testis, right? So that's regarding the tunica vaginalis. Here, you see the scrotal septum here, that's from inside, is dividing the scrotum to two cavities here, right and left. And pay attention to the anatomical position of the penis here. This side of the penis is called the ventral or anterior sides, because this is the anatomical position for the penis, right? Therefore, we say that the scrotum septum is attaching to the anterior side of the penis. And when we have the scrotum in the place, the fold that we have between the two is called raphe of the scrotum, as you see that here. Now let's see what other layers we have here. Besides the tunica vaginalis that we have, what other layer do we have in the scrotum? We have some muscles inside of the scrotum. The first one is called dartos muscle. Dartos muscle. Make sure you are differentiating dartos that you have inside of the scrotum from detrusor muscle. Detrusor muscle, where did you have it? In the urinary system, around the wall, in the wall of the urinary bladder. But here in the scrotum you have dartos muscle. So remember this. Then we have, this is a superficial smooth muscle, this dartos muscle, and when it contracts, it causes wrinkling of the scrotal surface. So in other words, when it contracts, you have it all around the insides of the scrotum. I will show it to you, and makes the scrotum wrinkle, and in other words, to get a little bit shorter and get closer to the body, right? Besides that one, we have another muscle that we call it cremaster muscle. Cremaster muscles that when contract, they tense the scrotal sac and pull it closer to the body. So as you see, the function of these two are similar, but that dartos muscle is right under the skin in the scrotum. This detrusor, it covers all around the testes and spermatic cord. Now the temperature for sperm development is 2 degrees Fahrenheit cooler than the body temperature, as we discussed. Therefore, if the temperature is cold outside, the testis moves closer to the abdomen by the use of these two muscles to maintain the proper temperature. If it gets too cold, also is not a proper temperature, right? So here you see this cremaster muscle, that it is insides of we have a fascia around that, we call it cremasteric fascia, and the muscle is cremaster muscle. Pay attention that these cremaster muscles is the continuation of one muscle that we had in front of the abdominal wall. Specifically, that is in continuation of the internal oblique muscle, right? Cremaster muscle. And also you have this layer of the detressor muscles. Let me see, it should be in the wall here. It has been shown here. Haven't been shown here, but that one is located here. Dartos muscle, this layer that you see, that is located in outer layer and pulls the whole scrotum upward. So that was regarding these muscles that you have inside of the scrotum and their function. And also, so the layers that you see, you have the scrotal skin, then you have the detressor muscle, then you have the fascia of the scrotum for the cremaster fascia, and then you have cremaster muscle here, right? So these are in the order. Now, you don't need to know these ones beyond the one that we discussed, all the artery and vein, as much as we have discussed before. We don't need beyond that. So for the structure of the testis, so that was regarding the scrotum, layers that it has and what they do. Now let's go over the structure of the testis itself. So we said that around the testis we have a layer, we call it tunica vaginalis. That is a serous membrane. That's a serous tissue that surrounds the tunica albugina. Tunica albugina, we're going to discuss about that. This is a layer that is located under tunica vaginalis, and that one is part of the structure of the testis, right? So tunica albugina is located under tunica vaginalis. When you open tunica vaginalis, that is around the testis, you can see tunica albugina. Now, tunica vaginalis is continuous with the surrounding tissue of the epididymis, covers around that, and it gets into the inguinal canal, right? And we said that this layer that is covering around the testis and epididymis is the visceral layer, and it continues when it gets to the neck of the epididymis. It looks like around and goes lines inside of the scrotal cavity, right? Now, if we open this one, what do we see? We see another layer that we call that tunica albugina. Albugina comes from alba, right? Although you remember that means white. This is a white layer that is a fibrous connective tissue, lining of the testis. It just goes line outside of the testis. And besides covering out of the testis, it sends some septa deep into the testis. It forms some septa, and between all these septa make some compartments inside of the testis, and those septa converge toward the mediastinum of the testis, the location that then it continues toward the epididymis. We call it mediastinum of the testis. Now mediastinum supports ducts, and those ducts move material toward the epididymis, right? Now let's go and see the picture of the testis, right? So here you see the scrotum from the outside, from the skin that you have dartos muscle, then you have superficial scrotal fascia, and then after that this superficial scrotal fascia, that's the one that we saw as the cremaster fascia, and then you have the cremaster, right? Then you see that you have the one layer of the tunica vaginalis is lining inside of the scrotum here, and then you have the other layers of that. After it gets here to this part, it goes around the part of the epididymis and then goes back around the testis, right? Now, under this other layer of the tunica vaginalis, you have this fibrous layer that is white here that we call it tunica albugina. That one is part of the structure of the testis. You cannot separate it because this one, as you see, it sends some septa inside, and as you see, makes some compartments here that you have a structure inside of that we call these tubes that are folded as the seminiferous tubule. Here you have seminiferous tubules. Seminiferous tubules are the tubules that are producing sperm. That we're going to talk about its structure, how the sperm is formed and everything, right? Then these tubes continue toward the network and finally they get into the epididymis here. So this area is called the mediastinum of the testis here, right? So these are the structure of the scrotum, all in the layer that you need to know in order, and these tunica albuginea and all these compartments that it makes, and the seminiferous tube. Now these seminiferous tubes continue to make when these end of these ducts come here toward the mediastinum, they make a network that we call it rete testis. Then this rete testis, so sperm is formed here in the seminiferous tubules, right? And in each compartment we may have 1 to 3, or mostly 2 to 3 seminiferous tubules. Then when they come here, they connect to the rete testis. Then after rete testis, you have very small ducts here that we call it efferent duct tube. Then after these efferent ducts, we have the epididymis here. Then after that, it continues as the vas deferens. The connection hadn't been shown because it goes down and then we continue to follow the path here. But you see one part of the vas deferens is coming and passing through here too, right? Now let's go to talk about all these tubules that are taking the sperm out of the testis. So again, regarding the histology of the testis, the septa, the testis separates, septa testis separate the testis into some lobules. So each compartment is called a lobule. Each lobule contains tightly coiled tubules that we call them seminiferous tubules. And each seminiferous tubule is approximately, if we unwind that, is approximately about 80 centimeter long. It's like 4/5 of the meter, right? So that's very long. And you see it has been coiled, it's tightly coiled so that it can be lodged over there, right? Now these seminiferous tubules begin to uncoil when it gets to the mediastinum of the testis and leading to the straight tubules. I think I didn't show those ones too. Those straight tubules in the mediastinum area then interconnect with each other, forming the rete testis. And within the mediastinum, we have the rete testis, then rete testis connects to the efferent ductus that I showed you, and then efferent ductus lead to the epididymis. So here these ones are straight tubules that we have, that they go from the seminiferous tubules to go toward the rete testis. Now there is a space between the coils of the seminiferous tubules, right? Like here, if you go back here, you see between all these ones, there is some space, right? That we have some cells inside of this space that we call them interstitial endocrine cells, or a famous name for them, is Leydig cells. So Leydig cells are the cells that are not inside of the seminiferous tubule. They are out of the seminiferous tubule, but they are inside of the lobule. So we call them interstitial endocrine cells or Leydig cells. Now these cells are producing and releasing the male sex hormone, the main sex hormone of the male, that is the testosterone. So that's important, and always remember that testosterone is produced by the Leydig cells. Other name for the Leydig cells is interstitial endocrine cells. Where they are located? In the interstitium around the seminiferous tubule, not inside of the seminiferous tubule. Now here, if you go back here, like if you look at this one, if you cut through like this, right? So you have the lobules that are surrounded with this septa, and inside of each lobule you have several of these holes of the tubules. So if you cut here, you will see several tube of the seminiferous tubule next to each other, right? So if we go over these sections that you see, like here you may see that one of them is horizontal, one of them, and some of them vertical, because they are located next to each other and coiled, right? And as you see, between those ones, you don't see the septa here. These are all inside of one compartment, the one have been shown here, right? So this is each tubule that you see here. This is one part of a tubule, another part of the tubule, another one that you see that here. And you see that out of each tubule, you have a space here. That's the one that we call it interstitium. And inside of this space is the location that we have the Leydig cells. Now, if we look at one single tubule, right, and you see that in a bigger scale, you see that you have a lumen inside and you have the surrounding part outside, right? Each tubule has a connective tissue around that we call it tubular capsule. Now we're going to talk about all types of the cell inside of each tubule, from the outside all the way toward from the capsule all the way toward the lumen. This seminiferous tubule, as we discussed, is the place that the sperm is going to be produced. And the direction of the production of the sperm starts from the periphery, goes all the way toward the lumen, right? Now let's see. So here you see that as we discussed, we have the hormone that is produced inside of the testes as testosterone. Now let's see what are the functions of the testosterone. Testosterone is not just for as the sex hormone, right? You will see that sex hormones also in the male and also in the female, they have many metabolic activity, and that's why when their level goes down, they affect many parts of the body, and the body starts aging like in a higher speed, right? Sorry. Now the functions of the testosterone. First of all, the main important thing, it stimulates spermatogenesis. What is spermatogenesis? What does it mean, genesis? Means production. Gen, as we discussed before, means production. Genesis means production of something. Production of what? Sperm. So spermatogenesis is the process that ends up to the production of the sperm, and it is stimulated by testosterone. The other function sorry, of the testosterone promotes the sperm maturation. The sperm is produced inside of the spermatocord, but later you will see that it is not ready to have its full functions that it has to be. It needs some maturation time, and that part is also under the promotion of the testosterone. The other function maintains the accessory organs. The accessory organs of the reproductive system are maintained by the testosterone. Develops the secondary sex characteristics. Those are the ones that we have after puberty, like that's the one that makes a difference between a child and an adult sense after puberty, right? Those secondary sex characteristics, mm, like the, for example, in the male, you have a change of the voice, you have all the building of the bodies, lots of muscles, and many other things, like the having the hair in some specific area of the body, like the mustache and all those things. Anything that is defining a human as, for example, here, a specific sex, like here as a male, those are secondary sexual characteristics, right? So those are developed by the testosterone. And then it stimulates growth and metabolism in general, and it stimulates sexual behavior and sexual drive. All those ones that are, those behaviors are necessary for reproductive system to be completely functional and serve its um, its main purpose and function of that, so that the two sex can be driven toward each other, and all those sexual behavior that makes the production of the zygote to happen. So these are stimulated by the testosterone. So those were the function of the testosterone that we mentioned. Now let's talk about the sperm and how the sperm is produced. As we discussed, we call that process as spermatogenesis. And spermatogenesis, it is a division that we call it meiosis. As you know, meiosis is the dividing of the cell and all its nucleus and everything, so that the final product, final cell, has half of the chromosome of the original cells, right? So let's see how this process is happening, right? So spermatogenesis is the formation of the sperm cell, genesis of the sperm, right? That begins in the outer layer of the seminiferous tubule. As you remember, it's a tubular capsule, right? Then the cells that we have in the outer layer of the seminiferous tubule, they called spermatogonia. Spermatogonium is singular, spermatogonia is plural, right? So each spermatogonium is a stem cells that gives rise to the sperm cells at the end, right? When we say stem cells, what does it mean? It means that it is able to produce more and more cells, and it is able to differentiate and develop to the other types of the cells, right? Now, these spermatogonia form during embryonic development. So before the baby is born, it's already have these ones inside of the testes. But then they remain dormant until puberty. So the baby is born with them inside of the testes. They do not have any function until puberty. They just remain as the way that they were until puberty happened, and then start going under more development, so that it ends up to the sperm formation, spermatogenesis, right? And just again defining the meiosis, that is a cell division that produces haploid gametes. Gametes, as we said, they are sex cells, and they have half of the chromosomes of the somatic cells. So somatic cells are considered as a diploid cell. The gametes, like the sperm and oocytes, they are considered as haploid cells. It means that when the cells becomes haploid, it has the half of the chromosomes of a diploid cell or a somatic cells. Each gamete has only one of each pair of the homologous chromosomes that has the, when it is a gamete, it is a haploid. it has a total of 23 chromosomes, not 23 pair of the chromosome that we have in a diploid cell that we have in a somatic cell, right? So that was the definition of the haploid cell, and remember that one because in the meiotic division, in the meiosis, we're going to discuss about that how it starts from the 46 chromosome cells and then finally ends up to a 23 chromosome cells. Now, spermatogenesis and meiosis. Beginning at the puberty, spermatogonia continuously divide throughout reproductive years. So this is one thing, just remember that this is happening in the male. In the male, we do not have the period. But sometimes you have spermatogenesis, sometimes you do not have it. There is no period, there is no pause. Continuously, spermatogenesis starts after puberty and continues as long as that person is alive, but the rate and the amount might decrease after the male person is aging, right? But still it is continuous. As each division occurs, meiosis division, one of the cells produced by this division remains in the outer, or is not meiosis. We first have a mitotic division. Mitotic division means that we have a spermatogonia. Some of those ones, like this spermatogonia, divides and makes in the mitosis. You remember that the result is two daughter cells that are identical completely to the original parent, right? So the first spermatogonia was 46 chromosome. The two daughters are completely identical to the parent spermatogonia, right? So one of the cells, one of these two daughter cells produced by this division remains in the outer layer of the seminiferous tubule in the original place as the stem cells, so that the pool of the stem cells remain intact. Otherwise, after a while, all the stem cells had gone, right? But from each division, one of them remains to just fill the place for the original parent, right? The other cell produced by this division differentiate to become a diploid primary spermatocyte. So the first one that goes into the process of the spermatogenesis is called a primary spermatocyte, and it is still diploid because it has 46 chromosomes. Right? The primary spermatocyte begins to undergo meiosis. For the meiosis, if you remember, we have two phases, meiosis 1 and meiosis 2. Meiosis associated with the formation of the sperm cell specifically is called spermatogenesis, as we said. The primary spermatocyte, that is the daughter cell that now is going under more development, undergoes meiosis 1 to produce two cells that we call them secondary spermatocytes, right? Now these, as you see, these primary spermatocytes get into meiosis 1. Now in the meiosis 1, the number of the chromosomes becomes half. Therefore, this secondary spermatocytes after meiosis 1 is haploid, right? So secondary spermatocyte is always haploid, right? We can switch it. The first one is double, the second one is half, right? Like that. So each one has one of each pair of the homologous chromosomes. That's a total of 23 chromosomes. Although each chromosome still has two sister chromatids. If you do not remember all these things about the meiosis, you have to go and review them in the division 1 and division 2 of the meiosis, meiosis 1 and meiosis 2, to make sure that you realize that, right? But that's the way that it is. They make a sister chromatid. Now the chromosomes get separated, each chromosome has still the sister chromatid with itself, right? But the number of the total chromosome is not 46 anymore. We have 23 chromosomes, each chromosome with one sister chromatid, right? After meiosis 1, the time that we have secondary spermatocyte. Then each haploid secondary spermatocyte goes under meiosis 2 and then produces 2 haploid spermatids. So after meiosis 2, we call that spermatid. The spermatid still needs to go under more maturation and change the shape to become a haploid sperm cells. I will show you the picture for all these ones and we'll review. Haploid cells contain 23 chromosomes. A single primary spermatocyte gives rise to a total of 4 sperm cells because it divides 2 times and all of them remain alive, right? That is another point that is different with the female, that later we're going to talk about the female. Over there you will see that each primary oocyte at the end gives you one only oocyte. But this one you see that each spermatocyte, primary spermatocyte, at the end gives you 4 sperm cells, right? So here, this picture showing all the spermatogenesis, right? So first we have spermatogonia, that is the stem cells. It goes under mitosis division. Then after that it has 2 daughter cells that each one is completely identical to the parent. One of them remains in the area under the capsule here, and then one of them goes under more development. Therefore, this one that goes under more development is called primary spermatocyte. That still is diploid, right? The same number of the chromosome as the parent. Then this one goes under meiosis 1, and after meiosis 1, it becomes half of the chromosomes, right? The number of the chromosomes becomes half. Therefore, each one of these primary spermatocytes makes the 2 secondary spermatocytes. Each secondary spermatocyte is haploid. So pay attention. Primary spermatocyte is diploid. Secondary spermatocyte is haploid. And then each one of these secondary spermatocytes goes under meiosis 2, and then those sister chromatids also get separated. So we have 23 here, 23 on the other one. So still each one of these 4 that come from these 2, and these 2 came from one single cell. Each primary spermatocyte makes the 4 spermatid, and spermatids are haploid, right? So these spermatids are not completely mature yet. They have to go under another process, more development, and each one of them make one sperm that is haploid, has 23 chromosomes. This maturation and this change from the spermatid to the sperm is called spermiogenesis. So pay attention, this last phase is called spermiogenesis, the production, the physical maturation of the sperm. The number of the chromosome is not changing, just the shape of the cell is changing. It loses some of the cytoplasm, becomes this shape, it makes one part that is made, makes the tail, and all those ones we're going to talk about this. This physical maturation is called spermiogenesis. So this is the final phase of the whole process, that is the spermatogenesis for the sperm production. Pay attention to definitions and the names. Right? Now, if we go and cut a part of the tubule, so you see that you have a tubule all around here. I can show only one part of that. If I go up and come back here, you see a circle that is showing a cross section. Now we just look at one part of that, and you see that this is the outer part of the tubule, that is a tubular capsule that you have here, right? Then you have from the periphery, you have spermatogonia, right? These ones that are in the outer layer. Then as they are developing, they move toward the middle and go toward the lumen. So in the process, spermatogonia makes the primary spermatocyte, then secondary spermatocyte, then spermatid, and finally the final ones are sperm. But they get into the lumen, and we have some fluid inside of the lumen of the tubule, and takes these sperms with itself to the next phase, like from the seminiferous tubule to the straight tubule, retrotestis, and all those tubules that we said to get out of the testis, right? And the other things that, before forgetting to mention, this is the space out of the tubule, between the tubules, that we call it interstitium. Therefore, these are interstitial endocrine cells or Leydig cells that they are producing the testosterone, right? Now, looking at this section of the tubule, we have a cell here that we call it nurse cell. These nurse cells, if you go over the borders of the cell, you will see that these are lodging all these cells of the spermatogenesis. So as you have the spermatogonium here, then you have primary spermatocytes, you have secondary. If you look at the border of these nerve cells, you see that this is the border. If I want to show you one single cell, you see the nucleus here, and now the border of the cells are these ones. This is one single cell that you see that here, all the way here with all these borders. So as you see these cells, it just provide a space for all these cells, and it's changing the shape as they are developing more and more, makes them more close toward the lumen, so that finally all these sperms that are produced, they get into the lumen and release from the cells and continue their way with the fluid that we have over there. These nerve cells have several functions that we're going to discuss about that, including, for example, production of the fluid that we have inside of the lumen. But before that, let me show you in this picture as I have it here. You see a dotted line here that it's showing the level of the blood testis barrier. You have the blood vessels around these capillaries around the tubule here, and you have a barrier between the material of the blood and the material that you have inside of the seminiferous tubules. Because if these ones get exposed to the blood, the white blood cells and the material that we have inside of the blood, they may start an immune reaction to these ones. Therefore, we have a barrier here so that they keep the border between these cells, between the material and the cells of the blood and these material of these cells of insides of the tubule. So just pay attention. This is the blood testis barrier that we have here. Now, regarding the spermiogenesis, as we said, this is the process that the spermatids mature to form the physically mature sperm cells. So this is not changing the number of the chromosomes, but it is changing the shape of the cells. And this maturation process is called the spermiogenesis. And during spermiogenesis, the spermatids are embedded in the nerve cells. And another name for the nerve cells are Sertoli cells. That's much more famous. So nerve cells are the same as Sertoli cells. Remember that one. Now, as we said, those cells remain inside of the nerve cells and go toward the lumen. And upon maturation, when it becomes completely, spermatid becomes completely mature, is called sperm cells, enter into the lumen of the seminiferous tube. So let's see the functions of the nerve cells. The function of the nerve cells is maintaining of the blood testis barrier. That's one function that isolates, as we said, isolates the seminiferous tubules from the circulation blood vessels. to prevent autoimmune destruction of the sperm cells, right? So that's one important function that is one of the jobs of the nerve cells or Sertoli cells. The other jobs support spermatogenesis, right? As we said, it gives them a place to be in there, to be organized and move toward the lumen. Supports of spermiogenesis at the end of the spermatogenesis. Secretions of inhibin. Inhibin is a hormone that controls the rate of the sperm formation, right? So this one can control that the sperm formation is be faster or slow. And other function of the nerve cell or Sertoli cell is secretion of the androgen binding proteins. Proteins that they can bind to the androgen. Androgen is a general term for the male sex hormones. One of them, for example, is testosterone. So these proteins are binding to the androgen. These binds testosterone within the seminiferous tubule so that you remember that will continue to have an effect on the spermiogenesis. Testosterone is produced in the Leydig cells that are out of the tubule. Now they bind to this protein, androgen binding protein, that are secreted from the nerve cells so that the testosterone can move inside of the tubule and do its function, the effect over the spermiogenesis and all the other things that we discussed, right? Spermiogenesis and all. Now here again you see that that's all the things that we already discussed. You are looking at one section of one single tubule here, and we just discussed about all these things. So that was regarding the spermatogenesis and how the stem cells, spermatogonium, changes and finally makes the sperm. Now, the structure of a sperm after the spermatid developed and make a mature sperm, each sperm that also is called a spermatozoon. A spermatozoon means one sperm, right? Pay attention, you may see that spermatozoa, Z-O-A, spermatozoa, that's a plural for the spermatozoon. So if you want to go over the structure of one single sperm or spermatozoon, it has three parts: head, middle piece, and tail. In some book you may see that they divide it to 4 parts, but they are all the same. They have put two parts in one as one. So the head of the sperm contains a chromosome. 23 chromosome of the sperm is located inside of the head as the nucleus, right? Contains acrosome on the top of the nucleus that consists of enzymes. So this acrosome that goes like a cap over the top of the nucleus in the head, it has the enzymes. Those enzymes, hyaluronidase, that is hyaluronidase, is necessary for the fertilization of the egg. It means that when the sperms reach to the ovocyte, it needs these enzymes on the top so that it can just make a hole inside of the wall of the ovocyte so that it can go inside, and then the other reaction that is going to happening after that, and finally the chromosomes of the sperm can be combined with the chromosomes of the ovocyte. So that's the function of the acrosome, and as you see, it's vital for its function of the sperm so that it can do the final job that is penetrating into the oocytes and combining its chromosomes with the chromosome of the oocyte. Middle piece of the sperm or spermatozoon, that includes the neck here. It contains mitochondria and centriole. So mitochondria, you know that is for the production of the energy, right? For the ATP production. And as the sperm is a cell that is moving all the time, if it is not moving, it is not functional, right? So it needs lots of mitochondria that are located in the middle. Besides of that, we have some centrioles in the middle piece of the sperm that includes the nerve. And finally, we have the end of the sperm that is called tail, and it is a flagellum. And that's the only flagellum that finds in the human body that we have. So if you remember from the chapter 2 that we discussed, that flagellum is the process at the end of that attaches to the cell and helps the whole cell to move, right? And the same is here. The whole sperm is moving by the flagella compared to the cilia that is moving on the surface of the cell and moves the surrounding material around the cells. But this one is moving the cells, and that's the one that is part of the motility movement of the sperm and enables mobility of the sperm cells. So here you see that how the spermiogenesis is happening. This is the spermatid. That you see it has Golgi apparatus, it has a nucleus, it has the mitochondria and some vesicles here. Now it shows that how these acrosomal vesicles, it just goes toward one end here, how it loses some of the cytoplasm, shed cytoplasm, becomes a slimmer, and then this flagellum is forming here, this next part is forming, and at the end you have this mature sperm with all these parts that you have a nucleus that contains the 23 chromosome, you have this acrosome as a cap on the top of that, it includes the enzyme hyaluronidase, and then you have the centrioles and mitochondria in this part that make the middle piece, and finally you have the flagellum that here have been cut because that's compared to the size of this part, the flagellum is very long here. And so that was the structure of the sperm, and here you see the real sperms by the electronic microscopes, all the parts here. So that was so far we talked about the sperm formation, how the spermatogenesis happens to finally from each spermatogonium you have 4 sperm or spermatozoa, right? So let's stop here and we continue the rest of that, how the sperm that has been formed in the seminiferous tubules gets into the rest of the tubules that we have in the male reproductive tract. We call those tubules as the tract, and then continue its way to get out of the body and get into the body of the female, to the reproductive system of the female, and then do its job to combine with the ovocyle here. So we'll stop here and continue the second video in the next one. Then see you soon.